In this video, I'll be solving paper 2 variant 1 May-June 2021 9700 biology. Figure 1.1 is a transmission electron micrograph of cells from duckweed Spirodella oligorhiza. Calculate the actual width of the cell labeled X. So you'd measure this width using your ruler. Write down the formula you will use to make your calculation and show your working, and give your answer in micrometers to 1 decimal place. So the formula you'd use would be actual size is image size divided by magnification, where the image size can be anywhere between 16 to 19 millimeters. To convert that to micrometers, you'd multiply by 1000 and then divide by magnification, which is 4275. And so your answer would be between 3.7 to 4.4 micrometers. B1 Table 1.1 lists some biological molecules found in plant cells. Complete Table 1.1 by choosing one letter from Figure 1.1 that indicates a cell structure where each biological molecule is found. DNA is found in nucleus, cellulose is found in cell walls, phospholipids would be found in cell surface membranes, and histone proteins again are found in are the protein molecules that hold together the DNA structure to bind the entire length in within nucleus. So first off, we have nucleus, that would be A over here, this entire structure, and then cell wall would be, if you zoom in to this picture, you'd see that the label E is for a cell wall. And then a cell surface membrane could be the membrane A over here, the nuclear membrane, or the membrane in the chloroplast of the thylakoid membrane, which would be C. And last but not least, histone proteins would be found with DNA in the nucleus, so the structure would be A. State the name of a cell structure visible in figure where ATP is synthesized. So that would be a chloroplast or mitochondria. These structures, which you see with lines, sort of like lines representing thylakoid membranes, are found in the chloroplast. So they synthesize ATP, which is why I'll write that down over here. 3. Name a structure of a cell structure that produces mRNA, and that would be the nucleus. C. Describe the evidence from figure 1.1 that shows that the image is a transmission electron micrograph. You've got 2 marks for this question, for this answer, and 1 mark I'd use to explain why it is an electron micrograph, and the other one for why it is a transmission electron micrograph. So first off, it is an electron micrograph because it has resolution enough to clearly show all of the organelles that it does in this micrograph over here. And also transmission electron micrograph are able to show the internal details of the organelles, which you can see for these chloroplasts, for example. These dotted lines represent hydrogen bonds. A. Describe the hydrogen bonding that occurs between water molecules as shown in figure 2.1. Alright, water molecules are dipolar, meaning that electron distribution in a single water molecule is not equal, and that happens because oxygen is a more electronegative element than hydrogen, so it attracts the electron bond pair in the covalent bond towards itself more than hydrogen does, and as a result oxygen gains a slight negative charge and hydrogen gains a slight positive charge. So the negative attracts the positive, and this results in formation of the hydrogen bond. And each oxygen atom of a water molecule forms 2 hydrogen bonds with hydrogen atoms of another water molecule. So those 2 you can see in the figure 2.1. B. The human enzyme salivary amylase is composed of 1 polypeptide. Figure 2.2 represents the structure of a molecule of silver amylase. 1. Explain the role of hydrogen bonding in maintaining the secondary structure of proteins such as salivary amylase. So secondary structure of proteins would be represented by alpha helix and beta pleated sheets, and hydrogen bonds are the only bonds that hold this structure. And so you can mention that over here. To explain the role of hydrogen bonding in maintaining the tertiary structure of proteins such as salivary amylase. The tertiary structure would be three-dimensional folding of the already folded polypeptide, and hydrogen bonds can do that as well when these bonds form between the CO group of one amino acid and the NH of another amino acid. C. Outline the importance of water as a solvent in plants. So water is used as a solvent in both of the transport vessels, phloem and xylem. In the xylem, it's used to transport the mineral ions, and in the phloem, it's used to transport assimilates. And the third mark, for the third mark, you can mention how it's also used to dissolve solvents inside the vacuole of plant cell. 3. Whisking tubing can be used to investigate the properties of cell membranes. A student carried out an experiment to investigate osmosis using whisking tubing. An outline of the investigation is shown in figure 3.1, where first you've got this whisking tubing measured against a measuring rule, and it's the meniscus which is used to measure the amount of sucrose solution present inside the whisking tubing, and there's a knot in the whisking tubing as well. And then that whisking tubing is placed in 15 centimeter cube of sucrose solution whose concentration is 0.9 mole per dm cube, and then the meniscus level is measured again. 6 pieces of whisking tubing were filled with 10 centimeter cube of different concentrations of sucrose solution, 0, 0.4, 0.8 and 1.2, 1.6 and 2 mole per dm cube. The height of the meniscus of each solution in the whisking tubing was measured. The pieces of whisking tubing were put into test tubes containing 15 cm cube of 0.9 mole per dm cube of sucrose solution. So you're changing the concentration of sucrose inside the tubing, and the concentration of the sucrose of the solution that surrounds the whisking tubing remains the same. After 20 minutes, the pieces of whisking tubing were removed from the test tubes, and height of the meniscus in each was measured. You've got the results in table 3.1. So as you increase the concentration of solution inside the tubing, the difference in height of meniscus after 20 minutes becomes more positive. The concentration of surrounding solution remains 0.9 mole per dm cube. So from 0 to 0.8 mole per dm cube, the concentration of solution inside the whisking tubing is less, and therefore it's got a greater water potential than the surrounding solution. So water moves out of the tubing, as a result of which when water moves out, the height decreases, and therefore the difference in height is negative. And from 1.2 to 2 mole per dm cube of whisking tubing solution, the concentration is greater in the tube than that outside, so water moves into the tubing by osmosis down the water potential gradient, and so the height increases. As the height increases, the difference in height becomes positive in the last 3 readings, and you're supposed to explain all of that in this answer. B. When red blood cells are placed in water, they are destroyed by bursting. The student also investigated how red blood cells are affected by immersion in solutions of sodium chloride of different concentrations. Blood samples of same volume were added to solutions of sodium chloride in separate test tubes. After 10 minutes, the student took 0.1 cm cube of blood samples from the test tubes and estimated the percentage of red blood cells that had burst in each blood sample, and figure 3.2 shows the student's result. Describe and explain the effects on red blood cells of immersion in different concentrations of sodium chloride as shown in figure 3.2. Alright, initially from 0 to 0.04 mole per dm cube of sodium chloride solution, the percentage of red blood cells destroyed by bursting remains constant, and then from that point onwards to 0.14 mole per dm cube of the salt solution, the percentage of red blood cells bursting decreases, and then it becomes 0 after the concentration of sodium chloride keeps increasing. Red blood cells burst when excess amount of water moves into them, and this happens when the cells are placed into a solution that has a greater water potential than the cells themselves. By increasing the concentration of sodium chloride solution, the water potential of the solutions that the cells are placed in decreases, and so water moves out of the red blood cells and into this solution. As this happens, the number of red blood cells that have excess water, or the cells that are going to burst, would decrease as you increase the concentration of sodium chloride. So that happens between 0.04 and 0.14 mole per dm cube of sodium chloride solution. From 0 to 0.04 mole per dm cube of sodium chloride solution, there are 100% of the red blood cells are bursting. That means for all of the red blood cells, the water potential of the surrounding solution is greater than the water potential of the cells, so water keeps moving in all of the 100% cells. And beyond 0.14 mole per dm cube of sodium chloride solution, none of the cells are bursting. That means none of the cells have excess water, meaning that water is moving out of them instead of moving into them, which also means that after 0.14 mole per dm cube, the water potential of the sodium chloride solution that surrounds the red blood cells is lower than the water potential of the red blood cells. You do not have to write the entire explanation. 4 marks is all you need, so you could distribute them evenly or however you want between the description of the graph and its explanation. For A, the induced fit hypothesis and the lock and key hypothesis are used to describe the mode of action of enzymes. Explain the induced fit hypothesis. So this happens when the active site is not entirely complementary to the shape to the substrate, so it moves around the substrate to provide a better fit and form the enzyme substrate complex. B. Radish plants contain the enzyme peroxidase that catalyzes the breakdown of hydrogen peroxide. Students investigated the effect of increasing the concentration of hydrogen peroxide on the activity of peroxidase extracted from radish, and the results of the investigation are shown in figure 4.1. So as you increase the substrate concentration, the rate of reaction is also measured. 1. Explain the effect of increasing the concentration of hydrogen peroxide on the initial rate of reaction as shown in figure 4.1. You could go with the usual answer for this type of question between the around the stage where the rate of reaction increases from 0 to 0.33 millimole per dm cube of hydrogen peroxide. The substrate concentration is the limiting factor, as there are more active sites empty than there are the substrate molecules, and so as the substrate concentration increases, the collision between substrate and active site also increases, therefore increasing the number of enzyme substrate complex forming, and therefore the rate of reaction also increases. After 0.033 millimole per dm cube of hydrogen peroxide, the substrate molecules are in excess, and now the limiting factor is the active site. All of them are now saturated, resulting in a constant rate of reaction, as the number of enzyme substrate complex forming is constant and not increasing as it initially was. The students determined the Michaelis-Menten constant for radish peroxidase as 0.1 millimole per dm cube. With reference to figure 4.1, describe how they determine this value. So Michaelis-Menten constant is the substrate concentration for which the enzymes work at half of their maximum velocity. So the substrate concentration you would obtain from the x-axis, but for that you need to know the maximum velocity, and so the half of the maximum velocity. Maximum velocity would be provided by the maximum rate of reaction, which is 5.6 micromole per minute of the rate. Half of that would be 2.8 micromole per minute of a reaction. And so you need to locate that on the y-axis. That would be just this one tiny unit below the 3, and the corresponding x-axis value for 2.6 units of rate would be 0.1 millimole per dm cube of hydrogen peroxide. See, a further investigation found that the Michaelis-Menten value of carbonic anhydrase is 12 millimole per dm cube. Describe the role of carbonic anhydrase in the transport of carbon monoxide. So this is an enzyme found in the red blood cells. It allows the reaction between carbon dioxide and water to form carbonic acid, and which then dissociates to form hydrogen ions and hydrogen carbonate ions. This hydrogen carbonate ion carries the carbon dioxide, which makes up 85% of the total carbon dioxide carried in the blood. 5. Much success was made in reducing the number of cases of malaria between 2000 and 2015. Explain how malaria is transmitted. It is transmitted by the vector female Anopheles mosquito when it takes blood meal from an infected person and then from an uninfected person. B. Diagnostic test strips from malaria can contain monoclonal antibodies. The test strips detect antigens produced by the pathogens that cause malaria. Figure 5.1 shows stages in the production of monoclonal antibodies. The information in three of these stages is completed, and you're supposed to complete the rest. Malarial antigen is injected into a mouse where the immune system responses, and this produces specific B lymphocyte cells that divide to form many plasma cells. And plasma cells are then extracted from the spleen of the mouse where they would have been stored. These plasma cells then fuse with myeloma cells, which are a type of cancer cells. This forms the hybridoma cell. See, figure 5.2 shows two diagnostic test strips for malaria. A sample of blood from a person suspected of having malaria is put into the well labeled S. A buffer solution is put into the well labeled A. The buffer solution moves the blood towards the results window. A line at position C indicates that the test is working correctly. A line at position D indicates a positive result for malaria. State 3 advantages of using test strips for malaria, such as those in figure 5.2. Alright, because the test does not differentiate between the 4 species of the pathogen that cause malaria, it can be used for all species of Plasmodium. Also, it would not be as expensive as other techniques might be, and it can easily be used. So those would be the 3 advantages I would be talking about. The highest number of cases of malaria occur in sub-Saharan Africa and Southeast Asia. Discuss the factors that determine the global distribution of malaria. A number of factors come into play in this answer. Warm and humid regions are favorable living conditions for Anopheles. Also, water bodies in such regions can provide breeding grounds for the mosquitoes that act as vectors. Mosquitoes can also develop resistance to insecticides, so eliminating them to break the transmission cycle can be difficult. Another fact is that Plasmodium, the pathogen that causes malaria, could develop resistance to anti-malarial drugs, and so all of those factors could cause difference in number of cases in different regions of the world. So in the regions where the insecticides no longer working on mosquitoes, or anti-malarial drugs become ineffective on the Plasmodium, the number of cases might rise. 6. Lysosomes are cell structures that contain enzymes known as acid hydrolysis. Figure 6.1 shows some processes that occur in animal cells. So here you've got bacterium engulfed into the cell by phagocytes. So this would be phagocytosis occurring over here. Arrow D shows how the bacterium is digested by the acids in the lysosomes. Also, the acid hydrolysis can be injected into worn out organelles like this mitochondria over here, so that the insides are digested also. A named the cell structures labeled A and E. So A is rough endoplasmic reticulum since it's got ribosomes associated, and E is the Golgi body or the Golgi complex. B state the function of the structures labeled F. Right. So as you can see, the structures F are also present on this side, and they bud off of the rough endoplasmic reticulum and then join the Golgi body. So these would be proteins synthesized. There would be proteins synthesized in these vesicles, and they are then transported to Golgi body from rough endoplasmic reticulum. D, with reference to the processes occurring at B and D in figure 6.1, outline the role of acid hydrolysis in lysosomes. Here you can see at D, the bacteria are digested so that it does not harm the cell, and the organelles are also digested. The only reason for it could be that the organelle is worn out, otherwise why would the cell damage itself? And so the question is of 3 marks, so you could provide in an extra detail about this cell. As you can see, the internal structure of the mitochondria that are usually shown in diagrams are cristae. So those are membranes made of phospholipids. So this enzyme could be digesting phospholipids over here. E, carrier proteins in the membranes of lysosomes maintain a lower pH than the surrounding cytoplasm by moving hydrogen ions. Suggest how the carrier proteins maintain the lower pH within the lysosomes. Lysosomes already have acidic pH due to having greater hydrogen ion concentration inside than the hydrogen ion concentration outside that is present in the cytoplasm. And to maintain this concentration of hydrogen ions, they have to keep moving hydrogen ions inside, and to do that against their concentration gradient, they have to move them by active transport. Alright, this is it. We are done with this paper. Thank you for watching.